Good afternoon, good evening, good morning, everybody. This is Elizabeth Room from the Netherlands, UPPMD, and I would welcome you all and also thank Prosensa for making it possible to have a webinar today where we can ask all our burning questions. The day that this meeting is as follows, this webinar. We first will have a presentation by Prosensa and by scientists. Dr. Campbell will be there and Dr. Craig McDonald as well. And you will unmute. You can send in questions in the chat box, although they won't be answered during this meeting. If you send a question to the chat box, you will get an answer later. You can also ask questions later. And at the end of the presentation by Giles Campion, Campbell, and Craig McDonald, you will hear the question you all send in and answers to that. So that is what we're doing today, and I would like to give Giles Campion, the medical director of Prosensa, the floor.
Thank you, Elizabeth, and I'd like to thank you also for the opportunity of giving the patient community an update as to the status of the Drisapersen program. As was indicated by Elizabeth, we have two guest scientists who will also be participating. Craig Campbell, who was involved in the phase 3 trial and subsequently in the extension study, and Craig McDonald, who's the principal investigator on the US dose-ranging study. Now, given that we're a public company, I have to give these forward-looking statements and then go on to talk about what we will cover during the course of this presentation prior to the Q&A. I just want to go through the recent events since the beginning of the year, what's been happening, and then Craig will be looking at the study 114673, which is the longest running study we have, over 177 weeks of observation on those trial subjects. And then we'll go through the 48-week results of the US phase 2 study, which Craig presented at the MDA clinical meeting last week in Chicago. Then I'll go on to talk about the intentions we have relating to redosing with Drisapersen, and also take you through some of the processes that we have to go through. And Craig Campbell then will also give his perspective from as an investigator. And then I will go on to cover the follow-on programs before finishing for Q&A. So this just gives an overview of the complete clinical program. It is comprehensive in scope, so over 300 patients have been involved in 3 placebo-controlled trials and various extension studies. And I say the ones that we will, Craig will focus on will be the Clino2, the one up on the top left-hand side there, 114673, and 117, and 876 down the bottom, the US study. Just to remind you of some of the results that have been reported with respect to these studies, either at the 48-week time point or the 24-week time point, and they show similar characteristics, that there is an initial rise from baseline. You can see in 673 of the order of 29 meters improvement from baseline, 35 meters in the 117 study, which is the European study at the primary endpoint, then 27 meters in 876 at the 6-month endpoint, and the phase 3 DEMAND-2, as you're aware, although there was a treatment benefit in favor of Drisapersen, this study didn't meet its primary endpoint. This just summarizes the key safety data that we've recorded across the 3 placebo-controlled studies, similar in that the most commonly reported adverse events include injection site reactions and renal adverse events, mainly consisting of subclinical proteinuria. There have been cases of thrombocytopenia which have been moderate severe in the longer-term extension studies. And Craig will talk a little bit more detail, in more detail regarding the US study. So just to recap what's been going on, at the beginning of the year, January the 12th, we regained rights to Drisapersen from GSK, and shortly after that reported some of the further analyses that had been done by that time, including some subset analysis, particularly looking at the those greater or less than 7 years old, the pooled studies, and also the extension study, particularly 349. There have been various communications with the patient community, both in January and then on February the 18th, after we had the IND, which was transferred from GSK to Prosensa at that date. Then last week, as I indicated, Craig reported the results of the 48-week study from the US in Chicago, and then following that, during our earnings call, we announced the intent to redose the initial group of boys by the third quarter of this year. So, starting off with the 48-week data, so this was new data. The 24-week data previously presented at the DIA and the World Muscle Society last year in September and October, respectively. And if you remember, the 24-week data was the treatment data, and this was followed up by 24 weeks off treatment, which was the 48-week data we presented last week. And Craig now will go on to discuss those results in the context of the other Drisapersen studies. So, Craig, if I can hand over to you now.
Great. Well, thank you very much, Elizabeth and Giles, for the opportunity to address the community. I'd like to start by updating the community with regard to the results from the long-term open-label extension study, where we actually have 177-week efficacy data. This data has previously been published. The earlier update has previously been published by Natalie Gomez in the New England Journal of Medicine, and the results are really quite impressive. Just to remind everyone about the original design of the phase 1/2 clinical trial, this was a dose escalation study followed by a 13-week follow-up period, and then the patients all initiated on drug at a dose of 6 milligrams per kilogram per week. There were 12 patients who initially were dosed. One actually was non-ambulant at the original onset of the trial, and another was non-ambulant at the time of this redosing here of 6 milligrams per kilo per week. And then I want to point out that actually the weekly dosing was actually converted over to intermittent dosing, where patients then received weekly dosing for 2 months, followed by a 1-month period where they were actually off drug. So essentially 2 months on drug, 1 month off drug. And when we look at the results here, they're really quite impressive with regard to the stabilization of ambulatory function, of walking function over the course of 177 weeks or 3.4 years. And you can see here there is really a number of patients who actually show improvement in their 6-minute walk distance. These are the 10 patients who were able to perform the 6-minute walk test. 2 of the patients who actually had lower values at baseline of redosing, values below 300 meters, actually subsequently went off their feet. They had values on the order of 240 to 260 meters. One patient actually with a lower value actually continued to show stabilization over a long period of time. And then the other patients, the majority had baseline values above 350 meters, and they've shown really quite impressive stabilization of their walking function over 3.4 years, with a median change in 6-minute walk distance actually of plus 7.5 meters over the course of the entire trial. And you can see that there seems to be a tendency of patients who are better functioning at the outset of the drug initiation to do better in terms of the stabilization of the walking test parameter. On the right, you see the ages of the patients. So they ranged in age from 9.3 to 15.4 years. All the patients were on stable doses of steroids, and all the patients were actually on the same dose of Drisapersen. I'd like to move on to the results from the U.S.-based study, initially discussing the 24-week study results and then the 48-week follow-up results. Just to remind people, there were 14 sites that conducted this study in the United States. The primary outcome measure was 24-week efficacy. Also, safety and tolerability was looked at, as well as other functional tests. Dystrophin data, and the key inclusion criteria were a baseline 6-minute walk distance of at least 75 meters, age of greater than 5 years, rise from the floor time initially of less than or equal to 7 seconds. There were actually 2 patients in the trial that actually subsequently had rise times greater than 7 seconds but less than 15 seconds under a modification to the protocol. And here's the baseline characteristics of these patients in this trial. I want to point out that they were all on steroids. Virtually all of them were on continuous regimens of steroids. And the baseline 6-minute walk values for the Drisapersen treated group treated with a dose of 6 milligrams per kilogram per week was about 396 meters over time. I want to point out this important distinction between 3 of the placebo-controlled trials that have been conducted by GSK and Prosensa. First of all, the U.S. trial showing the baseline 6-minute walk distance characteristics of 396 meters for the treated group, 416 meters for the placebo group. The study conducting in Europe comparing intermittent dosing with continuous dosing and placebo, again, the Drisapersen treated group in that study, 428 meters, 403 meters in the placebo group. And an important distinction in the pivotal phase 3 trial, the study 044, which was the pivotal trial conducted worldwide, the baseline 6-minute walk distance value in the Drisapersen treated group was much lower, 337 meters, whereas the placebo patients had a value of 348 meters. And this is an important distinction to make because baseline 6-minute walk distance actually predicts natural history and predicts subsequent disease progression. And just to point out, in the GSK 044 study, the pivotal trial, those who had in the placebo-treated arm, those who had a baseline value of greater than 330 meters lost 26 meters over the course of 48 weeks, whereas those who had a baseline value less than 330 meters actually lost 94 meters over the course of 48 weeks. So these are important distinctions and differences between the studies to point out. So the U.S. study actually evaluated 2 doses over 24 weeks initially with 48-week follow-up. Again, these patients were earlier in their disease progression. They could rise from the floor in less than 7 seconds. All but 2 of the patients enrolled in the trial, and a younger mean age of 7.6 years of age. Again, the baseline value for the treated group is on the order of 396 meters. In terms of the effectiveness data, this was the primary endpoint, the 6-minute walk distance at 24 weeks. And this really shows an impressive treatment effect fairly early by 24 weeks with an actual improvement in 6-minute walk distance and a treatment difference between the 6 milligram per kilo treated patients in the red, showing a 27.1 meter treatment difference in comparison to placebo in the blue, and the green is those on the lower dose of Drisapersen. I want to point out here that there has been concern in the community about the potential of the placebo effect due to injection site, potential unblinding by injection site reactions that may have occurred. And I want to point out that the patients treated at the lower dose of Drisapersen in the green there actually had equivalent injection site reactions to those that were treated in the higher dose, but yet they continued to show decline. So there really isn't, there does not appear to be any concern about placebo effect or unblinding by injection site reactions. And I also want to point out that a 27-meter change at 24 weeks is really quite clinically meaningful to a patient with Duchenne muscular dystrophy. So the next slide here, we show the actual percent predicted 6-minute walk distance changes, which really control for issues of maturation in patients that are perhaps growing or showing changes due to age and maturational effects. And this data actually shows the change in percent predicted value over 24 weeks. I want to point out that these patients had baseline values of 6-minute walk distance on the order of 70% predicted. So they were a less affected population, but it's really quite striking as those treated with the 6 milligram per kilogram Drisapersen dose actually show an improvement in their percent predicted 6-minute walk distance as opposed to those that were treated with placebo, which show a 3.5% decline. So a 5% treatment effect in terms of percent predicted 6-minute walk distance, which was almost statistically significant at the P0.51 level, even though the study was not really powered to demonstrate this treatment effect. I also want to point out that this change in percent predicted value by the placebo-treated group is very similar to what we've seen in natural history data as well. So in terms of the percentages of patients that show clinically meaningful changes, this is data showing the proportions of patients showing some improvement or more significant improvements of 30-meter changes or 60-meter changes. And if you look over there on the right, the Drisapersen treated patients, 72% of the patients showed some actual improvement in their 6-minute walk distance, whereas over 50% of the patients showed greater than a 30-meter change or a 60-meter change at 24 weeks. And at 48 weeks, that treatment effect seems to be persistent, with over 50% of patients showing either a 30 to 60-meter treatment effect over time. So this next slide actually shows the results of the follow-up post-treatment phase, so the 48-week follow-up in this U.S.-based study. And what's really quite impressive here is that the treatment effect seen at 24 weeks actually persists even after the drug is discontinued for 24 weeks. So there's a stabilization of disease which continues past the time that the drug is actually being dosed on a weekly basis, probably due to the long half-life of dystrophin. But really, I think the exciting aspect of this data really provides some validation for what was seen in the long-term 177-week extension trial, where patients were actually dosed on an intermittent basis, 2 months on and 1 month off, and actually maintained stability in terms of their disease progression over 3.4 years. So this U.S. data actually, I think, provides some scientific basis for the possibility of an intermittent dosing schedule in these patients. I also want to point out in these data, if you look at the bottom line there pertains to those on the 3 milligram kilo treatment dosing regimen. They actually did worse than placebo, but actually that was due to one patient who actually had a 200-meter decline at the 48-week endpoint. And so you can see, even with a study as robust as 17 patients in that treatment arm, 18 in the 6 per kilo treatment arm, one patient showing a change of 200 meters can skew the data that much. And so I think it points out the really quite substantial fluctuations that you get with trials of this size enrollment. The next slide actually shows the data controlling for maturational effects using percent predicted 6-minute walk distance And again, what it shows is a persistent treatment effect, which is maintained 24 weeks after the drug was discontinued. In the next slide, we again point to the adverse events, and what's really quite striking here, a high percentage of patients, both in the 3 per kilo and 6 per kilo groups, had injection site reactions. But in terms of renal abnormalities, proteinuria, or red blood cell in the urine, a very similar percentage of patients in the Drisapersen group, 28%, had proteinuria or red blood cells in the urine compared to the placebo group, where 31% showed those abnormalities. And in terms of post-treatment adverse events, there were essentially negligible post-treatment adverse events that were persistent to 48 weeks or 24 weeks after drug was discontinued. So any abnormality that was present at 24 weeks essentially went away over the next 24 weeks of treatment. So to summarize, there was a clinically meaningful treatment difference of 27 meters, an actual increase from baseline of 16.1 meters, which was observed in the 6 per kilo group compared to the placebo group. The difference was maintained 24 weeks after treatment was discontinued. For a percent predicted 6-minute walk distance, which actually controls for the issues of maturation, there was a treatment effect of 5.2% observed at 24 weeks. The lower dose of Drisapersen showed no clinically meaningful difference from placebo. Drisapersen was generally well tolerated, and the renal abnormalities in terms of proteinuria were actually fairly comparable to the placebo-treated group. This, again, was different than was shown in the European studies, but interestingly, than in the U.S. study, we showed fairly equivalent levels of proteinuria to the placebo group. The study really supports the use of 6 milligram per kilo once weekly in the treatment of boys eligible for exon 51 skipping, and the maintenance of treatment difference at 24 weeks was encouraging and consistent with potential slowing of disease progression in these ambulatory boys. And again, it brings the potential issue of intermittent dosing, I think, to the forefront for future studies. I want to then move to very exciting integrated analyses of these 2 phase 2 double-blind placebo trials, the European trial, which compared intermittent dosing with placebo, and then the U.S. trial, which compared the 2 dosing regimens. And if you really look here at this data, this is really quite impressive. Looking at those treated with Drisapersen for 6 milligrams per kilo per week, over 24 weeks, they show a 20-meter improvement in 6-minute walk distance, whereas the placebo patients actually showed an 11-meter decline. Again, the adjusted mean baseline values were on the order of 400 meters, but a treatment difference of 31 meters, which has been well characterized in the literature to be clinically meaningful at 48 weeks. But with this treatment, it's showing a treatment difference of 31 meters after only 24 weeks. And a p-value here of 0.003. And so, again, really quite statistically significant. And also pointing out here that these are all steroid-treated patients, and in terms of the placebo arm, these are all exon 51 skippable patients with the same levels of severity, all on steroids. And I think this, again, is very consistent with what we're seeing in the longer-term extension phase data. Then when we follow these patients in the European study, the 117 study, out to 2 years follow-up, again, to remind everyone, the baseline 6-minute walk distance values were on the order of 400 to 425 meters. And if we follow those patients who were actually treated for 2 years, 96 weeks essentially with Drisapersen, versus those who actually were treated for 48 weeks with placebo, followed by 48 weeks of Drisapersen, so a placebo-delayed treatment arm, and then comparing that with recently presented natural history data by Eugenia McCurry, which actually has focused on those aged greater than or equal to 7 and those with baseline values of greater than or equal to 350 meters. The results of Drisapersen are very significant. The 24-week outcome shows a treatment effect of 35 meters, statistically significant. The 48-week outcome was persistent. And then these patients followed for 96 weeks on Drisapersen only lose 5 meters, those 17 patients, whereas those patients that were in the placebo-delayed treatment arm lost 57 meters, 13 patients. Again, a very good placebo-delayed comparison for a treatment difference of 52 meters. So I think, again, when comparing with Eugenio Mercuri's data, shows a treatment effect of in excess of 70 meters. That's the data that Eugenio has presented that actually excludes those patients who lost ambulation. And when you impute 0 values, those changes in natural history data are on the order of 97 meters, including the 3 patients in Eugenio Mercuri's data set that actually lost ambulation. So with this, we'll turn the presentation back over to Giles.
Thanks very much, Craig. So turning from the recent data to now talking about the desire to try and get boys back on drug, the intent that we announced last week was to redose at least an initial group of boys in the 3rd quarter of this year. We are actively working on redosing plans for all the boys, for boys involved in all studies. But given the size of our company and the logistics, which I'll go through shortly, that will likely take a staged approach, either under a new treatment protocol or by an expanded access program, and that will vary likely country by country. And I do want to point out we understand that this is a very difficult period for boys and families, and certainly when I've spoken at meetings, this is a very constant question. But just be reassured that we're working as swiftly as possible to get this in place. And just going through some of the elements for consideration, the first thing that we have done, as you may be aware, is to get some indication of the numbers of patients wanting to redose so that we understand the logistics that we have to deal with. We need to understand the appropriate program to provide access to drug and get the required approvals from different regulatory authorities to do so. And then we have to ensure that we have sufficient clinical supplies to meet the needs of boys being redosed, both in the short term but also in the longer term as well. And importantly, there needs to be a prospective and acceptable regulatory path forward for Drisapersen. So in the agreement that we signed with GSK on the 12th of January in terms of transferring rights back to Prosensa, that involves transferring also all the data and supplies. As you can imagine, it's from the presentation, it's a complex and large data set. We have to make sure that all the information and data are transferred correctly so that we can then go out about being able to repeat the results that GSK have demonstrated. There's currently a joint transfer team of Prosensa and GSK working closely together on that, involving the transfer of data, know-how, and the drug product. And it really is a multifunctional activity, and you can see there in the diagram the different groups within the company that are involved. So the transfer process involves transfer of supplies of data, statistical programs, and results, and that's ongoing at this very minute. And just to give you some idea, I mean, just in terms of the statistical programs, these have to be rewritten. One, we have to verify the data that we've received. For example, there are 2 ways of writing the date, either with the day first or the month first, and we have to make sure that we've got the correct date format. It's a trivial example, but it gives you an indication of what needs to happen. Then the statistical programs have to be recreated, and then we have to regenerate the reports and tables and figures. And this is being done for each Drisapersen study, so you can imagine a lot of activity. Also, during this transition period, we need to determine where the clinical product supply is coming from, either in terms of the raw material that has been made, the drug substance, the drug product, which is what is sent to hospitals in vials, and then the process of packing and distribution for clinical trials. And I want to come back now to the survey that was sent to investigators, and I'd like to hand over now to Craig Campbell, who will comment on this activity.
Yeah, thank you, Giles, so much for inviting me to participate in this, and thank you to all the families who have come forward to be here. So I have been very interested, obviously, in strategies and mechanisms for redosing, and with Prosensa, I've been interested in looking at the survey responses. And if people aren't aware of this, on February 18th, a survey was sent out to investigators asking for feedback about the willingness of participants to go back onto redosing regimens. This would have been shared with you, or hopefully have been shared with you, in the context of closeout activities at your site. And I'll just say right here, because I was looking previously at the questions that are coming in, and one person asked specifically about this, so I may as well answer it here because I think it's most appropriate. I think if you haven't heard about this survey and haven't been able to provide some direct feedback to your local investigator, I would encourage you to contact them and assure that they have your response, but also to assure that they have submitted back that survey, because this is really important for planning purposes and design of redosing programs. And I know people will say, well, of course, everyone wants to go back on the drug, but that's not stringent enough documentation. And so I think it really is important to participate in this survey response. Right now, the response rate is suboptimal. The response is back so far about 60% from the sites, and of those responses, there certainly is an overwhelming majority of participants who have indicated that they wish to be involved in redosing. That's 90% of participants have indicated that they're willing to do so. 7% have said they're not willing to do so, and 3% are undecided. And we're also getting some comments back about why people might be unwilling or willing, and we'll be looking at those as we go through in more detail. But right now we still are looking for many sites to send in the responses. But generally, the responses have been very positive, not only in terms of willingness to participate, but also just in terms of the experience in the clinical trial. One thing I will say, and this came up in a question as well that's come through, this is participation in the survey is not seen as a final decision to participate or not in a redosing program. So you can answer that survey honestly, and those will be based on the scientific analysis of the studies and mechanisms in place for redosing. So please follow through on that. And then I'll go to the next slide here. I think probably no one's bugged Prosensa and Giles with different scenarios and possible mechanisms for redosing as we have here in Canada. So I appreciate the time they've taken to walk us through. And I will say here that there is a very genuine and thoughtful approach to this on the Prosensa side, and but there are lots of things to deal with as we go through this process. And I think Giles has just spoken to the first of all that difficult task of transitioning back the data and the responsibility of the previously completed studies back to Prosensa, and so that's a large task. I think what you'll see on the slide here is a bunch of things we've put in, just very pragmatic things that we need to think about as we plan for redosing. But even a lot of these things that are up on the slide are still yet downstream from some of the pillars that Prosensa is working on with the DMD community and the investigators. And so that includes regulatory discussions, and people will have been aware of comments in all the Prosensa communications that they want to do this in a way that's responsible to the regulatory pathway of this product and products that are still in the pipeline for DMD, and so it's very important to work carefully with the regulatory agencies. I think we see that the various regulatory agencies, particularly in the U.S., have said they're committed to finding paths forward for novel therapeutics in DMD, and so it's important that we work with them to find that proper way forward that doesn't jeopardize either this program or others in the pipeline. Of course, the other component is that there's different mechanisms for redosing in different areas of the world, and so plans may come in different permutations, different flavors, if you will, and those all have to be sorted out. And of course, that's all in the context of managing the company and the drugs that are still in the pipeline for the company. And of course, as I mentioned, I definitely get a clear indication that keeping the patients and the patient community at the forefront in terms of communication and information has been quite important to Prosensa. And then, if things go the way of actual reinitiating clinical trial, then these are all the components you see on this slide, including reconstituting data safety boards, getting sites together and organized ethics committees, approvals, et cetera, et cetera. So there are lots of steps to move through to make sure that a redosing plan is viable. So I'll stop there and let Giles move through the next phase of the presentation.
I guess Giles will come soon, but...
Yeah, sorry, I was on mute. I have to apologize for that. Craig, I just wanted to thank you, and I was also saying that going through these various steps is not by way of an excuse, but really trying to help everybody understand why redosing can't start tomorrow and why we're having to work very hard to get through these activities, but that we're totally committed to being able to find a way forward. The next part, I just want to address the other programs because there were a number of questions submitted ahead of this call relating to other DMD programs. I mean, clearly, it's been very important to understand the results of the Drisapersen program, particularly the phase 3 study, and Craig has been taking you through some of the thoughts regarding that. But clearly, we want to make sure that the follow-on programs are optimized as far as they can be in the light of the results that we've seen. So just going into some of the specifics, Pro44, the phase 1/2 study has been completed there with evidence of dystrophin restoration. The dose modeling that we did to look at exposure predicts that an effective dose range will be between 6 and 9 milligrams per kilogram, and we're going to do an intravenous program with this particular compound. Safety findings have been consistent with the known class safety profile, so similar to what we've seen before. No drug-related serious adverse events. Of course, the IV formulation should deal with injection site reactions. And the start of the extension study is expected somewhere between towards the second half of this year, but again is dependent on the final analysis of Drisapersen. The next program, we have two programs ongoing in dose ranging. One is Pro45, so looking at individuals who need exon 45 skipping. The idea here is to use the natural history study subjects as controls, and I'll talk briefly about that. The results of the phase 1/2 dose escalation study are expected in the second half of this year, and the expanded treatment phase, which will follow on, again will be dependent on the final Drisapersen path forward. 53 is also in the clinic in dose escalation phase. The study results here will be expected again towards the end of the year, and we will, as with all the other programs, rely on our total understanding of the Drisapersen data to optimally design that final phase. Programs skipping exon 52 and 55 are in the preclinical stage, and again dependent on Drisapersen. We also have a program, have been very cognizant of the issue of rare mutations, and so we have a program looking at exons initially between 10 and 18 or 10 to 30. This is a technique that allows the skipping of multiple exons with a single antisense oligonucleotide, so has advantages in terms of developability. Proof of concept has been obtained in multiple patient cells, and we should be seeing selection of the best candidate somewhere near the first half of this year. It's also a technique that can be applied to those individuals with more common deletions as well, and again would have the advantage of being able to deal with a number of mutations with a single drug. Finally, the natural history study. So we're now well through that. Currently 190 patients are enrolled, and the idea of this is not only to understand the disease better, but also to follow new biomarkers that we're looking at to be able to assess the effectiveness, the usefulness of imaging, and also we hope to use the information from this study to help provide control subjects for our programs with rare exons where having enough patients to put on placebo becomes very difficult. So this is another very important program to us, and I think really signifies our commitment to the whole disease area. So finally, I want to thank everybody. We want to thank all the boys and their families that have participated or are participating in the clinical programs. We really understand the commitment and sacrifice that it has to be made to participate in these trials. and we understand the distress in terms of wondering when redosing would start. We'd like to thank the community at large for their input and their patience, and we're continuing to work with clinical experts such as both of our guests, the regulators and patient groups on the next step. And we really are committed to doing what is in the best interest of the Duchenne community to Drisapersen and our other programs. So with that, I'm going to turn it back to Elizabeth, who's going to coordinate the Q&A session.
Thank you so much. For the people who tuned in later, if you felt it was really too fast, although I have to thank the speakers for speaking slower than they usually do, the same slides will be available on the website of Prosensa, and we will announce when they are available there. So if you feel you missed something, you do have a lot of opportunity to read it on the website. I will now read the question you sent ahead of the webinar to the parent project, the different patient organizations. And the first question is one I would like to direct to Giles Campion, and that is, when will you begin redosing the boys who participated in the clinical trial?
Thank you for the question. Tried to address that during the course of the talk. We did indicate last week that we were aiming to redose in the third quarter this year, and the steps that I outlined are the ones that we're currently engaged in to be able to complete that.
Right. Thank you. And then the next question also to you is, can you tell us something more about a drug protocol for redosing these boys, and when to expect announcement of this protocol?
Again, something we tried to address in the presentation. I think that, as you know, this was a global program, so there are many different countries involved, and there are different programs which make most sense to particular countries. So at the moment, that's what we're looking at to see what program would suit which country. There are protocols that we are considering for specific jurisdictions, and we'll be discussing those with the investigators involved in the coming few months.
Right. And do you plan to use the same clinical trial sites?
Yes, that is currently the intention. I think one thing that we're always aware of is the issues that patients and their families face in the travel they have to do. So there were some attempts previously, I know particularly in the US, to try and find more local sites. So that's something that we would continue to look for. But I think that the key thing there is to try and get redosing started as soon as we can.
And is there adequate supply of materials to redose some or all?
Yes, I mean, that is not a hurdle at the moment. As you can imagine, GSK were making plans for the potential commercialization of this product, so they had been a manufacturing program in place, so material supply is not an issue at this moment.
Right. And the next question I would like to address to Craig Campbell. It's a question who came in. I heard that the redosing survey is really important, but my physician at the trial site has not contacted me. What should I do? Will my son be left out of any redosing plans? I think that is one you can address again.
Yeah, and I did try and tackle that during the talk as well, but definitely the redosing survey is important. As I mentioned, it's although maybe one's first impression is to say, oh, we've all called our investigator, talked to them, said that we're interested in redosing. Unless it's, clearly documented, it's not as helpful to Prosensa to feed back to regulatory agencies or in their plans for redosing. So I think it is important to be in touch with your investigator, get a confirmation that they have heard your response, and they are planning or have done the submission of that survey. We're looking at those carefully. So please do that if you can. It's helpful not only for yourselves but the other boys who are involved at your site. But having said that, the survey will not be used as a strategy to discount or discard sites or discard boys from the possibility of redosing. That is not the intent of the survey. So not to panic if your site doesn't manage to get that in. So either individually or by sites, that is not the intent of the survey at all.
Thank you. Another question to Giles Campion is, what progress was made since the last webinar?
I hope I've convinced you that we have actually been working very hard, both in terms of everything that needs to be done to transfer all the material and data from GSK to us. You can imagine we're a relatively small company, and our activities up until that point were primarily to do the work we've been doing on the other compounds apart from Drisapersen. So we were relatively abruptly handed back the rights of this large program. So we've been having to work very hard to make sure that we can take this on board and find an appropriate path forward for the boys and their families.
The next question is about pulmonary function. I would like to address it to Craig McDonald. The question is pulmonary function looked at for Drisapersen? Will there be trials for Drisapersen or other exons with somewhat older boys to look specifically at the effects of pulmonary function?
That's really a great question. I think pulmonary function is a really important endpoint. It's one that has to be generally looked at over longer-term trials, and so I think we're in a position, certainly with the patients that have undergone the 177-week follow-up, as well as those that have been followed now into 2 years, to look at pulmonary function. And there's plans to actually evaluate for stability of pulmonary function vis-a-vis the available natural history data, using the appropriate prediction equations. And I think it's also important to point out that even those patients that perhaps were more severely impaired at the initiation of treatment in terms of the ambulatory measurements, that doesn't mean that this drug still could not be having significant effects on stabilizing upper limb function or pulmonary function. And so this is an exceedingly important issue to address, and I'm sure the company will be focusing a lot of effort on it in the coming months.
Thank you so much. Another question to Giles Campion is, a family is asking, our son is 5.5 years old. He's suffering from deletion of exon 45 till 50 out of frame. Is there any chance to put him on the list for clinical trials with Drisapersen? Do you intend to extend trials with younger boys between 5 and 7 years old? And if yes, when would it be possible?
Well, thank you for the question. I think what we aiming to do first is to try and give individuals that have already been on the trials with Drisapersen access to drugs. So I think that's our first priority. In terms of the particular age group, we have, as you've heard from Craig, boys of that age have actually figured quite prominently in our clinical trials. So it is certainly an age group that we have studied extensively. And I think in terms of additional trials, that will depend to some extent on the conversations we have with regulators. So I can't make any predictions at this point in time.
Thank you. And a question to Craig McDonald is, given the phase 3 results, has a sub-analysis provided insight into these results?
I'm sorry, could you repeat the question?
Sorry. Given the phase 3 results, has the sub-analysis provided insight into these results?
Yeah, I think it's safe to say, with regard to the sub-analyses, that patients on this form of dystrophin restoration therapy, it seems that patients, in terms of an ambulatory endpoint, those that have higher baseline levels of function seem to actually respond more to the drug. They even show improvement in 6-minute walk distance over the short term. I think that as we look at those that are showing that have more severe impairment at the initiation of treatment, I think what we're seeing is that there are subsets of patients that show quite substantial decline, whether they're on treatment or placebo, that were really I think destined to show decline. And when you have a large change of, say, 200 or 300 or 350 meters in a given patient, that can really impact the data quite substantially. So I think one approach to deal with that is to actually do longer-term trials. And I think by all indications with the subgroup analyses, I think that the Drisapersen treatment effects seem to be more substantial as you start following patients out to 2 years study duration. And I think there has been an impact of some of these patients with lower levels of functioning at outset, which is not really too different than was seen with Eteplirsen in their experience. The two boys with the worst levels of functioning were those that went off their feet very abruptly. So I think there has been a lot of insight, and there's going to be additional sub-analyses, I'm sure, over the coming months of this data.
Thank you. Maybe Giles Campion can ask this question we received, saying, personally, I would like to try this medication to see if it makes any difference, quality of life-wise, and potentially to stop the progression or even reverse some of the damage. What is the chance of getting this medication to market and to the consumer soon?
Well, thank you for the question. I mean, as you can imagine, we are totally committed to being able to find a path forward for Drisapersen as quickly as we can. And I think as you know, there has been comments made by the regulators and specifically the FDA highlighting Duchenne muscular dystrophy as a condition which has the highest priority for them. So we're working hard to try and find a path forward and get to market as soon as we possibly can, if that is feasible.
Okay, thank you. Greg Campbell, can you ask the question? Can muscle that are lost be repaired by exon skipping? I think they mean muscle cells.
Right. So yeah, I think most people who are on the conference here would recognize that the process in Duchenne muscular dystrophy is that over time the muscle cells themselves, the muscle fibers, are struggling to stay healthy and eventually get to a point where each individual muscle fiber is not able to be viable anymore or to stay alive anymore, and then those muscle fibers are replaced by either fat cells or fibrosis, essentially a kind of a scarring tissue. So as with most of the therapies directed at Duchenne, dystrophin restorative therapies like exon skipping really require a a viable muscle cell in which the dystrophin that is then being reconstituted, theoretically by the drug, would be integrated into that muscle fiber to provide it the stability it needs, and reverse that process, or at least stabilize that process. So that's theoretically how we think about this. Of course, with the exon skipping, the dystrophin protein is changed into, again, the theoretical situation is it's changed into a dystrophin protein that's still shortened. It's not complete, but it has some of the major components that allow it to be reintegrated into the muscle cell. And so in short, no, muscle fibers, muscles that are lost, to our knowledge, would not be corrected through exon skipping. And I think that is perhaps what we see. Dr. McDonald just talked about how some of those kids who were in a phase where they were rapidly losing ambulation, that was hard to reverse or maintain in the studies, even if the drug we feel is effective. And so muscle fibers that are in jeopardy and muscle fibers that are lost may not really be able to be repaired by exon skipping.
Thank you. And then there's a question again for Giles Campion. Somebody asked, I would like to know if you will be starting a trial for exon 53, and if you are, when will this trial begin? Thank you.
What I indicated in the presentation that we are currently doing a dose ranging study, and we anticipate the results of that towards the end of this year. We do have plans to then recruit more patients into a treatment phase. Again, how that study will look will depend on the final analysis of the Drisapersen study. So I can't say exactly when that would start at this moment.
Thank you. And then to Craig Campbell, did changing the age range from 5 till 13 in the phase 2 studies to age 5 to 16 in the phase 3 study lead to the loss of statistical significance?
Yeah, that's a good question. I mean, I think we've spoken to some of these things in the presentation, in that certainly the older children would be expected in general to have lower 6-minute walk tests, would be perhaps in a group more in jeopardy of losing ambulation, and so those things may well have factored into some of the difficulties or differences anyways between the phase 2 results that were shown and the phase 3 results. I guess in general, one always has a struggle in larger clinical trials, in the sense that the more patients one enrolls and the wider the inclusion, the more variability there becomes in the study, and then that does have an impact on understanding statistical significance. If there's more variability in your study population, then it can become more difficult to see the effectiveness signal amongst all that variability. I don't know if Craig or Giles want to add anything to that.
Yeah, I think it's important to point out that perhaps more so than the age range of the study, I think that the study 044 really did include a patient population who was on the average more severely affected than the other studies, and I think that's been demonstrated both in terms of baseline 6-minute walk distances and then baseline levels of other functional evaluations such as time to rise and other parameters. But again, these are all clinical design issues and issues in terms of adequately powering a clinical trial. I think that doesn't mean that there isn't a subset of population that, in terms of an ambulatory endpoint or an ambulatory measurement, could really be showing benefit to the treatment. And it also doesn't imply that this drug might not be helpful to those with more severe disease that perhaps have lost the ability to ambulate. This drug potentially could be affecting skeletal muscle and help stabilize upper limb function as well as pulmonary function and other parameters. But to demonstrate that, it will take different trial designs and possibly longer follow-up, possibly even registry-based follow-up long-term.
Okay, and then somebody asked for an update on ongoing programs 44, 45, 53. And do you have plans to extend the studies in other countries? And if so, what countries and what is the timeline?
Yes, as I indicated during the presentation, we do have plans to carry on with those developments, but we want to make sure at the same time as we analyze the Drisapersen data that we do optimize the program. I mean, many of the elements that Craig has been talking about, the importance of baseline 6-minute walk, the condition of the boys when they go into the study, are clearly important in terms of predicting outcomes. So we want to make, before we start further programs, we need to make sure that we have really optimized the trial design to do everything we can to ensure a successful outcome.
And the next question to you is, test results show that Drisapersen works best if treatment is started early in young boys. Does this mean that other trials, for instance exon 53 skipping, that is relevant for our son, will also focus only on young boys from now on?
Well, the answer to that question is that no, the intent of the program that we are planning to run is that it will not only focus on a particular age group of boys, but will also include those individuals who aren't able to walk anymore. And the rationale for that was actually articulated by the FDA in a meeting just before Christmas, in which they said that as long as you defined the group where you expected to see efficacy, they actually encouraged the enrollment of other boys as well, accepting that ambulation is only one potential endpoint. So the intent of the follow-on programs is to be broader than just a specific age range.
Thank you. And then the last question, and I'm sure you will say I covered that partly as well already in the presentation, is our son has a deletion of exon 48 till 52. When will there be a drug for exon 53, and will it also become available for boys 16 years of age?
Yes, well, the program for 53 does include a plan to enroll non-ambulant boys, so I assume the question is directed towards involvement in clinical trials rather than a drug becoming available commercially. And clearly the goal of all of our programs is to get drug available commercially to boys as soon as possible. So that's definitely in our plans.
Okay, thank you so much. This is then the end of our question and answer session. The question you sent to the chat box will be answered later, and you can also, you see the email addresses on the slides. You can keep sending questions. So what is left now is to thank Prosensa for sharing their data and future plans with us. I also would like to thank everybody calling in and thank the families who participated in trials. And a big thank you for the people at the PPMD office who really make this webinar technically possible. Thank you so much, and till the next call.